Welcome everyone to the 8th and also season 2 premiere of Contact's Produce Safety webinar series. The second season will continue to bring together industry, academia, and regulatory to solve some of the produce industry's biggest and most timely challenges. This series is part of a larger industry outreach and risk mitigation project called Contact, or scientific challenges and cost-effective management of risks associated with implementation of produce safety regulations. This project is supported by the Specialty Crops Research Initiative from the USDA National Institute of Food and Agriculture. Any opinions, findings, conclusions, or recommendations expressed in this presentation are those of the speakers and do not necessarily reflect the view of the U.S. Department of Agriculture. This webinar series, coupled with other research and extension efforts, is part of a collaborative effort across 10 different universities or government institutions to enhance produce safety knowledge and compliance across the industry. We are so grateful to our collaborators for their efforts and support of this webinar series. You can see their logos on the screen. This webinar series will continue next month with a talk by Dr. Keith Schneider about post-harvest water management on Thursday, October 13th. You'll have an opportunity to register for this webinar after the conclusion of this one. This year's webinar series will address brand new topics chosen by you, the listeners. Other than the topics shown on the screen, we've got an exciting new list of webinars scheduled for this season covering topics like sanitation, foodborne outbreak attribution, traceability, and more. If you think a topic is missing from this list, let us know in the comments, webinar surveys, or on our social media pages. While this webinar series lets us meet with you once a month, that's just not enough for us. We'd love to #make contact with you about today's webinar or other produce safety news and research on our Twitter and Instagram pages. Follow us on these platforms at PS_Sci. Be sure to check out the Contact website for more produce safety resources, including our key takeaway documents from each webinar, also webinar recordings or other featured content from this series under Webinars tab on the website or on our YouTube page. You can find these at the links on the slide or go to the Contact homepage, click on the drop-down menu and select Resources or Google Produce Safety Science YouTube. A quick reminder for today, if you would like to submit a question for our speaker during or before the Q&A session, please submit your questions through the Q&A box and not the chat box. Now, let's welcome our speaker today, Dr. Leanne Jaykus. Dr. Leanne Jaykus is the William Neal Reynolds Distinguished Professor in the Department of Food, Bioprocessing, and Nutrition Sciences at North Carolina State University. She served as the Scientific Director of Norovirus. The collective was a large consortium of scientists and stakeholders working collectively to reduce the burden of foodborne illness associated with enteric viruses. Her professional activities have included membership on the National Advisory Committee on Microbial Criteria for Foods, and as the president of the International Association for Food Protection, IAFP, in 2010 to 2011. She has taught food microbiology and safety courses, mentored over 60 graduate and postgraduate students, and authored or co-authored over 200 publications. Dr. Jaykus received a BS and MS degrees in food science from Purdue University and a PhD from the University of North Carolina, Chapel Hill School of Public Health. Welcome, Leanne. The floor is yours.
Good afternoon. I am delighted to be here to talk to you about enteric viruses and contamination in soft fruit with a focus on berries. And I love this term, management of risk in the face of uncertainty. I hope you come away with the fact that there's a lot of uncertainty here. By way of an outline, I'm going to give you a little bit of a background on the viruses that we're concerned about and how they can enter the food chain, including some information about the epidemiology of outbreaks associated with soft fruits and berries in the last maybe 3 decades. I'm then going to move on to contamination routes and then spend quite a bit of time talking about the FDA Berry Sampling Plan and the associated methods, testing considerations, and impacts that has had on growers and processors. And I'm going to wrap up with some discussion about prevention and control strategies, and then happy to take whatever questions you have at the end. So let's get started and talk about my favorite subject, which is human enteric viruses. Human being from people, enteric meaning associated with the gastrointestinal tract. This particular group of viruses is what I call a functional rather than a taxonomic group. What that means is that they have similarities in structure, function, infection, transmission, et cetera, but they actually are represented by a wide variety of different virus families. Now, viruses in general are what we call obligate intracellular parasites. What that means is that they require a live host cell, a specific host cell that they can actually infect in order to replicate. So they basically enter the cell, they use the cellular machinery to make more viruses, but they can't do anything in the absence of that cellular machinery. So they are really not alive. And I'm always very careful to not use the word kill when I talk about getting rid of viruses, so to speak. I rather use the word inactivate or make them non-infectious. They're very different than bacteria and parasitic protozoa, which I hear you guys have had some webinars about. Very simple structure. First of all, they're about 1/10 to 1/100th the size of a typical bacterial cell. And if you look on the left-hand side of this slide, there's a cartoon of a norovirus right here. They're extraordinarily simple structure. So the virus actually consists of an outer protein coat that we call a capsid. And inside that protein coat is a piece of viral nucleic acid. DNA is nucleic acid, okay? Although in this case for the viruses, it's a single strand of RNA, which is the genome or the molecular or the genetic material that carries all the information about the virus. These viruses have no lipid envelope. They're called non-enveloped viruses. And that's important. Some really important viruses that you're aware of, I'm sure influenza, you know of, certainly COVID, both have lipid envelopes. And that is really important because that lipid envelope tends to be pretty sensitive to a lot of disinfectants and sanitizers. So that's one of the absence of a lipid envelope is one of the drivers for the fact that these viruses are so difficult to inactivate. They are transmitted, and this is extremely important, they are transmitted by humans only, specifically by the fecal oral route. So they're shed in the feces of infected individuals. That fecal material ends up in the mouth, goes through into the GI tract and establishes infection. Now, there is an exception to that. In vomitus or emesis for norovirus infections, virus is also shed, and that can also serve as a potential source of contamination. And the other thing I want to tell you about, there are multiple transmission pathways for these viruses. So we talk about the quote foodborne viruses, but really the most common way many of these viruses are transmitted is by contact between people. So I'm going to limit most of my discussion here on foods, but keep in mind that they're transmitted in many other different ways. So I can't go into the details. I can do a whole presentation on how viruses are unique, but I'm going to skip that and give you the key points that you need to keep in mind as we go through this presentation that talks about management of these viruses in the food chain. They are extraordinarily low dose organisms. What that means is it doesn't take much to make people sick. And it depends on what you read and who's done the study. But the estimate is somewhere between 10 and 1,000 particles is necessary to make an individual sick. And I tend to use the number about 100 when I talk or think about these viruses. With that said, they're excreted in extraordinary high concentrations in the fecal material of an infected individual. And we're talking millions per billions per gram. And so if you do the math and you have 100 infectious dose and millions in a gram of fecal material, you can see that even a tiny amount of fecal material has hundreds, if not thousands, of infectious doses of these viruses. Because they require a host cell, they're inert in the environment and they're inert in foods. They cannot grow outside of that host cell, nor can they grow in foods or the environment. With that said, they're extremely environmentally stable. If I were to put a small amount of norovirus on a stainless steel surface, it would survive and be able to initiate infection after as long as 4 to 8 weeks of sitting on that surface. So extremely stable, much more stable than bacteria. And they're resistant to many food processes, preservation, sanitizers, disinfectants that are routinely used in the food industry. So they are one tough nut to inactivate. Because these viruses in particular, the ones we're going to talk about, cannot be readily propagated in vitro. In other words, they can't really be easily grown in the laboratory, nor is there an animal model outside of human beings. So when I work with noroviruses in the laboratory, what I tend to do is get fecal matter from an infected individual. That's my source of virus. And then I have to use molecular biology techniques because I can't grow it in the lab. I have to use different type of technique to detect. This actually leads to one of the central tenets of this presentation, which is that these guys are really difficult to detect, and it's very difficult to interpret detection results. And we'll get into a lot more of the nitty-gritty of this as the presentation moves along. But bottom line is you can't grow them. You got to use molecular methods, and the relationship between molecular detection and the presence of an infectious agent is tenuous at best. So these are the major enteric viruses of epidemiological significance that tend to have some common food transmission source. And the ones I'm going to really focus on today are hepatitis A virus and human norovirus. Rotavirus is the leading cause of infantile diarrhea. It's a real problem in developing countries. Good news is we have a vaccination strategy for it. And this is a virus that we have greater control over, particularly as we have increased vaccination in our population. Hepatitis E is an emerging virus, which we really don't talk about a lot in the US, but the Europeans are quite concerned about it, actually. So in terms of Hep A and Noro, which is what I'm going to talk about today, they come from two different virus families, and they cause very different diseases and very different potential disease manifestations. And their prevalence in the population is also very different. One thing before we launch into a little more detail about each of these viruses, I want to bring out is that hepatitis A virus in the United States is a reportable disease. That means if somebody is diagnosed with Hep A, their data is delivered to the CDC, and oftentimes their clinical specimens also go to the CDC. So CDC keeps track of hepatitis A cases around the country. Norovirus is not a reportable disease. So we have much less information on what's happening in the norovirus world. And it should be pretty clear to you why this is the case. So a little bit about hepatitis A virus, very long incubation period, which is a problem when it comes to looking at food attribution, because people oftentimes get Hep A and they have no idea what they ate 4 weeks ago. Its typical hepatitis symptoms start with a sort of malaise and fever, and it then moves on to classic jaundice, darkened urine, painful liver. And usually in most cases, the disease runs its course and people will be fine after they've been infected. It is very different than hepatitis B and C, which can cause chronic active hepatitis. This virus tends to not do that. It is by far the most serious of the foodborne viral infections. In kids, young kids, probably less than 8 to 10, almost completely asymptomatic. A young child will not even know that they're sick. But severity increases with age, and overall hospitalization and mortality rates are relatively low, as you get to higher ages or a more aged population, you begin to see much more hospitalization and much higher mortality rates. And that's why when you see an outbreak of Hep A, almost always they also report hospitalization and death rates. The good news is immunity is permanent, whether you get infected and you become immune or whether you become vaccinated. And we have had a vaccination available now for almost 30 years. This virus is largely transmitted by either fecally, orally, mostly by male sexual relations, but also can be transmitted by IV drug abuse. And so only about 5% of Hep A cases in the country actually come from foodborne routes. So norovirus, on the other hand, completely different. And I love this slide because it totally explains what norovirus is. This is the virus that my mom, when I was growing up, called the 24-hour bug, where you have vomiting and diarrhea and you feel like you're going to die, but you don't die and you recover in a day or two. It is also what is fondly referred to as the cruise line virus. With that said, cruises are a very minimal contributor to the burden of norovirus disease. It is worldwide the leading cause of acute gastroenteritis, very low hospitalization and mortality rates. You almost always completely recover from this illness. When you look at all transmission routes, there are somewhere between 20 and 25 million cases of norovirus in the United States every year, which results in a reasonably large number of hospitalizations and deaths, not by virtue of an elevated rates, but by virtue of the fact that there are just so many illnesses that are occurring. Now, if you just take out foodborne transmission of noroviruses, that's about 20% of the cases, which equates to somewhere around 5 million cases a year associated with consumption of contaminated food. The hospitalizations and deaths estimated by, I think this is from Elaine Scallan's work, are provided here. This places norovirus second in total food-related hospitalizations, only second to salmonella, and it ranks noro fourth in deaths. So I want to talk a little bit about the major foods that are implicated in norovirus outbreaks, and then really dig much more deeply into the whole berry or soft fruit issue. So there's 3 general categories that are prone to or more likely to be contaminated with viruses, both noro and Hep A. The first is, and really the most important, are what we call the ready-to-eat foods. And I'm actually using this in a very distinct category, that being foods that receive a lot of human handling just prior to consumption and without any type of terminal processing or heating step. Okay, so think sandwiches, think salads, that kind of thing. That is probably 70% of norovirus outbreaks, and the same for foodborne hepatitis A outbreaks are associated with those food products. The second category are fruits and vegetables, and the third category are molluscan shellfish, so oysters, clams, etc. So as I had mentioned, noro's not a reportable disease, so it's very hard to get a feel for attribution relative to outbreaks. But Aaron Hall and his colleagues at CDC about 10 years ago did a study using some of the data from the National Outbreak Reporting System, and they looked at what foods were most often associated with foodborne norovirus outbreaks, and if the location or point in the chain could be identified as to where the contaminant entered the food product, that being either during production and processing, during preparation and service, which really is retail environment and homes, or unknown. And fruits and nuts, and this is together, fall into kind of the second most prevalent category for causes of foodborne norovirus outbreaks. Keep in mind that for fruits and nuts, large proportion, at least a third, we have no idea where the contaminant or how the virus got into the food. For about 40% of those outbreaks, we know it came in through preparation and service. That's generally associated with poor hygiene of infected food handlers. And then there's a very small amount that's been attributed to production and processing. But speaking, this preparation and service seems to be a major driver in outbreaks associated with fruits. And that's an important piece of information to keep in the back of your mind as we talk more about this. So let's now really turn our attention to fresh produce. And you guys, I'm sure, are all familiar with the 4 W's, those being water, workers, waste, and wildlife, keeping in mind that norovirus is not transmitted by animals. We can take wildlife off that list. And so the 3 major routes for berries we would be concerned about would be, did the virus come from water? Did it come from workers? Or did it come from wastes? So fecal oral transmission, you got to have the feces from an infected individual. Those can enter the water supply in a number of ways. One is untreated or poorly treated sewage. And the most common way that happens is sewage leaks comes from flooding events. So when you have massive flooding events, sewage treatment plants become overloaded and you get either poorly treated sewage or untreated sewage going into the water column. Leaking septic tanks, which can certainly contaminate any kind of underground water or surface water, depending upon where the leaks occur, and direct defecation in fields. And actually, there have been instances where particularly children have defecated in fields and were infected with Noro or Hep A, deposited their fecal matter, and that has contaminated the product. So the second way that contamination can happen has to do, and we mentioned it before, with poor personal hygiene of food handlers. So in the berry supply chain, the most common way that would happen before you hit the retail sector would be either pickers in the fields who have not washed their hands after using the restrooms, but they have a norovirus infection, or packers in, for example, if you're doing field packing or if you're doing further processing, such as IQF for frozen berries. There are 2 papers that came out relatively recently. They're excellent, excellent review papers looking at contamination, control, outbreaks associated between enteric viruses, specifically Hep A and Noro, and berries. And I'm going to show you some data from the Boskert article, the upper one, to illustrate some of the points that I'm going to make in the coming slides. So this is the first table in that paper. And what they did is these authors went through all of the outbreaks globally that had been reported between 1983 and 2018. And this table represents the synopsis of those outbreaks for hepatitis A virus. And each table, they go into details about when it happened, the type of berry, whether it was frozen or fresh, where the outbreak occurred, where the product originated, because sometimes the product originates from a different place where the outbreak occurred, the source of contamination, if it's known, and the food product. And when you have time, and these slides will be made available to you, when you have time, it would be great to peruse these slides and get a feel for historically where we've seen berry-associated outbreaks of hepatitis A. A similar table was produced for norovirus outbreaks in this same paper. These authors also went back and they looked more globally at the data and put a few figures together, and I'm showing these to you for illustration purposes. The figure on the left-hand side is the country of origin for contaminated product, and the blue is hepatitis A, and red is norovirus. And you can see it's a wide range of countries in which the contaminated product originated. The graph on the right shows you number of outbreaks versus berry type for HAV on the top and norovirus on the bottom. And you can see here that for HAV, you have more outbreaks associated with strawberries and berry mixes. For norovirus, you have more outbreaks associated with raspberries and strawberries. What I wanted to do was put some of the basic concepts together and themes together from these two review articles for you to get a landscape for what's going on here. So clearly there are more berry-associated viral outbreaks occurring in Europe than there are in the US and Canada, just from a numbers standpoint. In addition, and this is extraordinarily important, the majority of outbreaks are associated with frozen berry products, not fresh berries. In addition, the majority of outbreaks are associated with product that was produced in other countries and exported to another country, oftentimes used as an ingredient in a blend. And so there are certain countries or certain regions of the world from which we have a higher or greater history of having received contaminated product. And those origins consist of Eastern Europe, the Middle East, China, and certain areas in Central and South America. Another kind of common theme for these origins and the contaminated product that comes from these origins is that they're almost always produced on very small farms, usually less than 10 hectares. And what happens is those small farms come together and they commingle their product, and then the commingled product goes to a distribution center for wherever it's going to go from there. Outbreaks are often small. There have been some large outbreaks of particularly Hep A in the United States, for example. I think 2 that I can think of off the top of my head in the last 30 years, but it's usually less than 50 cases. Traceability and the ability to find the contamination source are real challenges. So it's often the contamination source is often not identified, particularly for norovirus outbreaks. When it is identified, it is oftentimes a downstream food handler in the food preparation phase. And that goes back to Dr. Hall's work at CDC, where he clearly showed that it appeared that a lot of product was getting contaminated in the preparation phase, not so much in the growing or processing phase. So this is a slide that shows just a few of the norovirus and Hep A outbreaks that have been associated with berries and berry products in the past. I could do whole presentations on these outbreaks, and they are fascinating studies. But the 2 berry ones on the right were associated with fresh berries. Both of those occurred quite recently within the last 5 years. The ones on the left were associated with frozen product, and those occurred less recently, really probably within the last 10 years. So what I did when I put that first slide together, and I thought, I'm going to go in and see what the berry-related viral outbreaks were in the United States in a 25 to 30-year period of time. And if you look for our country in frozen berries, between 1990 and 2016, there were 4 hepatitis A outbreaks that resulted in 600 illnesses. And the details of those outbreaks are shown below. Between that same 25-year period of time, there were 3 norovirus outbreaks that were associated with about 150 illnesses, all frozen product. When you look at the fresh berry product, much less. So in 2019, there was a small outbreak of 20 cases, and in 2022, we're just getting out of that outbreak that was associated with fresh organic strawberries. So a lot less outbreaks for fresh berries. If you take that and move it more globally to the rest of the world, between 1983 and 2018, there were 68 reported berry outbreaks. And again, the vast majority, 50 of the 68, were associated with frozen berries. The last point I think is critical, and it's important to note this, even though there haven't been a lot of outbreaks or a lot of reported outbreaks, more than 80% of berry-related foodborne outbreaks between 1983 and 2018 were caused by viruses. So if you get a berry-associated outbreak, it's going to be more likely to be caused by hepatitis A or norovirus. Which leads me to a little bit of a discussion about epidemiological attribution. So attribution really refers to, can you attribute a particular product to a particular burden of disease? And unfortunately for norovirus, we can't because we don't have good stats. But we do, since Hep A is a reportable disease, we do have good stats for Hep A. So on the left-hand side here, I pulled off from a recent CDC report the trends in hepatitis A infections in the United States from 2012 to 2019. And the pink line you'll see is actually the reported cases. The green bars are estimated infections. That takes into account underreporting. And you can see here that the numbers of Hep A cases in the United States from a reportable standpoint usually has been somewhere around 1,500 a year. And even taking underreporting into account, probably never more than about 3,000 a year. And then all of a sudden in 2017, we saw this huge uptick. So the first thing to say, or the first thing to note is the relative importance of food to these outbreaks between 2012 and 26, while it's unknown, are likely to be pretty low. I think we said like about 5% of cases are associated with foodborne infection. And of those, berries are a minimal contributor. Most infections or foodborne Hep A infections occur in restaurants because of infected food handlers. This big peak, and this peak is actually getting larger, to be honest, is associated with Hep A transmission largely in the homeless population. And it is driven by increases in homelessness, mostly because of increases in drug addiction. And this peak has absolutely nothing to do with food. I'm going to jump into the Berry Sampling Program. This is conducted by the Food and Drug Administration. It was initiated in November of 2018, so about 6 months before the pandemic started. And the intention of this program was to get a feel for what the natural prevalence of contamination of frozen berries was with either hepatitis A virus or human norovirus. And the intention was to sample 2,000 different products, half of those domestically produced, half of those of imported origin. And samples were collected largely from distribution centers and warehouses. Again, testing for these two viruses. And testing was done using PCR followed by Sanger sequence as a confirmation step. And we'll talk a little bit about those methods in a second. And if FDA detected a fragment of viral nucleic acid or a fragment of the viral DNA, then the agency took some sort of regulatory action. And we'll talk again about that in a second. So I mentioned before that these viruses cannot be cultivated in the lab. They can't be grown in the lab. Those of you who are familiar with how we detect bacterial pathogens in foods, we use this little flow diagram I have here, which includes enrichment steps where you grow the pathogen, you grow the bacteria to higher levels so that it becomes more detectable, followed by plating, where you isolate the bacteria on a plate, and then you go into confirmation steps. You can't do these first three steps with these viruses. Instead, what you have to do is you have to concentrate and purify them out of the sample matrix. You go from a sample of 25 or 50 grams down to a sample that has maybe a milliliter with concentration of the virus and removal of the food product matrix. You follow that by nucleic acid extraction to get the nucleic acid out of the virus, out of the sample. And then you go into PCR, or we use reverse transcription quantitative PCR for viruses. A hugely important thing to realize in this is that at the end of this process, there is no culture. It is not like a bacteria where you can do whole genome sequencing, you can strain type it in the traditional means. You don't have a culture. You are simply making a decision about whether that product is contaminated based on the presence or absence of a very small piece of nucleic acid. That's it. So there are a number of developing detection methods for viruses in foods has been a pretty active area of research for the last probably 30 years. It actually was one of the topics that I studied when I did my dissertation work at UNC Chapel Hill. But two methods for detecting viruses in soft fruits have risen to the top, so to speak. And the first method is the ISO method. It's an international standardized method. It's been very well validated, and it's used more or less globally. And just to show you the complexity of this method, you begin with your sample, you add a small amount of an enzyme that breaks up some of the sample matrix and also allows the viruses to be released. You then get rid of the solids and you have a fluid suspension in which there's virus and a little bit of residual food matrix. You then use something called polyethylene glycol, which is a protein precipitate, to precipitate or allow those viruses to settle. You end up with a much smaller volume of sample that has the viruses in them. It's further clarified by solvent extraction, and then subjected to nucleic acid amplification and RT-qPCR. Okay, this is the sort of the international standard method that's used in most places in the world. The US Food and Drug Administration has a separate method that they use to test for viruses in soft fruits, and it differs from the ISO method. And so I'm going to quickly run through this flow diagram. Again, you start with your sample and you add an enzyme to break down some of the sample matrix. You then go into an ultracentrifugation step, which is very high speed spinning, which allows the viruses to settle and sediment into a very small volume. That's then followed by a solvent clarification step, a much more extensive RNA extraction method, and then the sample goes into the detection platform, which is RT-qPCR and interpretation of results. There are other countries in the world that have their own protocols. Most of those protocols followed the ISO method, which was the previous method we thought about. There is an exception in that Canada has just produced a completely different protocol that they are using for managing this situation in their country. So what are the major differences between the ISO and the FDA methods? Sample size is a big difference. So ISO is a 25-gram sample, FDA is a 50-gram sample. The virus concentration methods differ. One's PEG precipitation, one's ultracentrifugation. The RNA extraction or the complexity of the RNA extraction differs. The ISO one is a little less prescriptive and it's a little simpler than the FDA method, which requires multiple extraction steps. And then there are also differences in things like the positive controls and the process controls and criteria for determining sample positivity and negativity and the necessity for confirmation. And then you can add to that a bunch of minor differences that probably don't make that much of an impact on the methods, but there are also a number of minor differences. Unfortunately, we have never done a head-to-head comparison of the FDA and ISO methods. So we do not know how comparable they are in terms of assay sensitivity, assay specificity, all of those kinds of things. And equivalency of methods is very important and needs to be established sooner rather than later. And a major reason equivalency needs to be established is because most of the commercial labs out there in the United States only offer the ISO method. So we don't know how results from the ISO method compared to results you might get from the FDA method. I mentioned that there's a lot of complexities in virus testing methods, and I have spent the last 3 years doing many presentations about this. I just want to give you guys a sort of synopsis of this. As we mentioned, choice of method is an issue. If you're getting commercial testing done, it is going to be the ISO method because that is what the commercial labs offer. If you're getting sampling or regulatory testing done in the United States, it's going to be done by the FDA. So they are going to use the FDA BAM method. The whole process of concentrating and purifying the viruses from berries before detection is complicated, it's expensive, it's time consuming, it takes highly trained personnel, and it's not efficient. Usually you will lose up to 50, sometimes 90% of the virus that was there. That's going to impact or reduce your detection limits, make it harder to detect. And sometimes you have residual matrix. And residual matrix is incredibly important because it will interfere with your downstream detection method, which is a PCR-based method. So we're concerned about these residual inhibitors because they often require us to dilute the sample even before we test it. Another concern with any PCR method is that it tends to be prone to cross-contamination. It's easy to get one single amplicon into a tube and end up with a false positive result because of that cross-contamination. So you always have to be very careful and very aware of that. And of course, there are huge interpretation issues, which I'm going to go over in a second, in terms of how do you interpret a potentially positive signal and what is the relationship between that and risk to public health. I'll talk a tad bit about confirmation. We really do confirmation to do two things. One is to rule out cross-contamination, and the second is to identify or try to identify the strain that's present in the sample. And one last thing I forgot to tell you guys about the commercial testing is that it's expensive too. And so it's highly unlikely that you would be able to get commercial testing done for less than $200 to $250 a sample, which makes doing massive testing pretty cost prohibitive. So this slide I put together to explain a little bit about PCR amplification and why this is important in terms of interpretation of results. So I apologize for those of you who know a lot about PCR. I want to segue a little bit and explain this method. So PCR targets a specific sequence of, in this case, the viral RNA or the viral gene. And it amplifies that specific sequence in an exponential manner. So we target that sequence, and one copy of that sequence becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, and so on and so forth, until after you've run enough PCR cycles, you have millions of sequences. And we can calibrate and make this at least semi-quantitative by looking at our endpoint, which is something called the CT value, and the concentration of the viral gene that we're trying to detect. And that's called a standard curve. And the rule of thumb is that the higher the CT value, the less viral RNA or less viral genome is present. Okay, T values fall between 20 and 45. So CT values in the 40 to 45 region, you're going to have very small amounts of viral RNA there. The important point here is that PCR is detecting genetic material from the virus. It is not detecting infectious virus. So when you say, I have a positive test result for a PCR test result for norovirus, what you have is a positive PCR test result for the presence of noroviral RNA. That's all you have. You have no idea if that virus is infectious. And we are now know and are increasingly getting information on the fact that nucleic acid is pretty stable in the environment. It sticks around for a long time. And the take-home message is that just because you find detection of viral nucleic acid doesn't necessarily mean you have infectious virus there. And if you don't have infectious virus there, you don't have a risk of disease. So this is just illustrative of how in naturally contaminated samples, how high these CT values generally are. This was a surveillance study that was done in Canada, and they pulled all the positive samples out and looked at what the CT values of the positive samples are. And I just want to point out, you have a lot of them that are in that 40, that 39, 40, 41 range, which is very low copy number, not a lot of viral genetic material. So what has happened over the course of the last 3 years relative to the FDA frozen berry sampling program? The current status as of July of 2022 is that over 1,100 samples have been collected. About a third of them were domestic and about 2/3 were of imported origin. There were 5 positives, 4 of which were sequence confirmed for Hep A, and 10 positives, 7 of which were sequence confirmed for Norovirus. And the details of where these samples came from and percentages are provided on the right-hand side of this slide. The sampling plan was halted in probably June of 2020 because of the pandemic. It has restarted in August of this year, and the FDA will no longer be collecting a total of 2,000 samples. It will be more like 1,500 or 1,600. They have about 425 yet to collect and test, which they're in the process of doing right now. This leads to a conversation about why or how do we interpret these results. I can tell you from the early days of the sampling plan, any positive sample that was sequence confirmed resulted in a recall. Despite the fact that we don't necessarily know the relationship between infectivity of the virus and PCR signals, there's been some discussion that the presence of viral RNA in a berry sample is a microbiological indicator. And an indicator is simply a harmless proxy for a particular condition or environment that may have impacted a product. This concept of the presence of viral RNA is an indicator for human fecal pollution or human fecal contamination. And this is my opinion, but I don't believe that the data is out there to support using a PCR positive, particularly at very high CT, as an indicator of potential human pollution or human contamination of a food product. Number one, it's a tiny fragment of DNA that you're amplifying, usually only about 1/100th of the genome of the virus. Number two, there is no science that has yet been produced that shows a specific correlation between PCR and virus infectivity. We know nothing about the persistence or prevalence of small pieces of DNA or RNA in the environment. And I'll tell you, my environmental virology colleagues are in the process of trying to get to the bottom of this, because there is some belief that maybe there is a lot of pieces of nucleic acid floating around in the environment that are completely harmless. So all we can really say at the present time is that the presence of a viral RNA fragment amplified by PCR may or may not represent contact with human wastes. This is a really interesting paper that just came out and is the only thing that I've seen in the literature to date that even remotely investigated is a product that tests positive for virus also going to be associated with illness or cause illness. And this was actually a human challenge study that was done in China, where they had people buy berries. They tested the berries for the presence of viral contamination, and they looked to see if people got sick from eating those berries. Take-home message was, out of 120 consumptions, no one got sick. Although about 40% of those samples, the samples tested, showed some evidence by PCR of viral nucleic acid presence. So this is a really good example of the fact that these products tested positive but didn't cause disease in the consuming public. And in point of fact, even all of the products that were recalled in association with the sampling plan, not a one of them was associated with any kind of reported disease. And some of that product was on the market for anywhere from 2 to 8 months prior to being tested. So what has been the impact of this to the industry? Well, I don't really have to go into details here. There have been economic issues, certainly cost of reputation. In the early days of the sampling plan, there were lost sales, and that has not been sustained to date, but I have no idea what's going to happen in the future. I do know that FDA has approached AFI and may have approached some of the stakeholders who are on this call as well about putting a food safety prevention and control plan in place specifically for the safety of berries. The results we have to date are all, remember, associated only with frozen berries. but the prevention and control strategies would be similar for frozen versus fresh. And so this will be something that will be in development over the next year, probably. So what are some of the considerations if you're going to manage potential contamination of berries, either during production or during processing? And keep in mind that it's likely that the large outbreaks, probably product does get contaminated during the production phase. Simply, it would be very difficult to contaminate massive amounts of product during IQF processing. But the kinds of things that you would consider would be training and certifications that farms might have. Irrigation type and source is very important. Keep in mind, you want your irrigation and any water that's used for pesticide application, for example, to come from sources of water that are not impacted by human fecal pollution. And harvesting. If you do mechanical harvesting, you don't have fingers touching products, so you have much less risk of contaminating a product with viruses than if you had manual picking. And in IQF processing, really, it's a similar kind of thing. You want to pay attention to your water as well as personal hygiene and exclusion of ill workers. So potential risk factors that could be evaluated in a prevention and control strategy would certainly all focus on workers, water, and waste. I want to make the point that in order to really put a good prevention and control strategy together, you need to have a root cause analysis. If this product is truly contaminated with viruses, where did the contamination come from? At this point in time, we have not done that. I would make the argument that root cause analysis needs to be done so we can find where, if indeed contamination is coming into the chain, where it's coming in. But some of the potential strategies for workers could be vaccination. We have a Hep A vaccine. There's Noro vaccines in development. I have no idea when they'll be released, nor do I know how efficacious they'll be. Vomitus cleanup, which is done pretty routinely now in the retail food sector, and certainly paying attention to personal hygiene and hand washing. From a water standpoint, make sure your water sources are clean and monitored. And from a waste standpoint, consideration of these endemic regions where we have historically seen contaminated product coming from. The other major issue is if you're going to put in a good mitigation strategy, you have to have lot traceability, and that lot traceability has to be very reliable. And at this point in time, that is complicated for the berry supply chain. Finally, I want to talk a little bit about testing for verification purposes. It may be an option, but keep in mind the FDA method is not performed by most commercial testing labs, and it's going to be expensive to use testing as a verification. So my conclusion, certainly both fresh and frozen berries are considered ready-to-eat products, and we have to come to grips with that. These are ready-to-eat products. Historically, the risk of domestically produced berries causing an outbreak is very small. Likewise, the risk is very low for fresh berries, at least right now in our country. Outbreaks are largely driven by imported products, usually coming from small farms, endemic regions, where product is commingled among multiple small farms. The sampling plan has already resulted in about 10 recalls and significant impacts to the industry. Certainly, we already talked about reliance on just the FDA method complicates using other methods for verification purposes. Once the sampling plan is completed, we don't know what the implications of those data will be. Will berries be considered a product of elevated risk? I don't know. How are we going to implement prevention and control strategies if we don't even know where the contaminant is coming from? Finally, particularly given what's happened with fresh berries over the last couple of years, will we soon be faced with similar types of sampling plans and or considerations of management plans in the fresh berry chain? And I think that's probably likely, although I don't have evidence that will be the case. I could only give you a snapshot of the complexity of this issue. There are tons of other issues associated with this that I can't cover in this presentation, but I'm happy to talk about in Q&A. And finally, and this is incredibly important, new territory is being charted as regulatory actions are taken on non-cultivable pathogens detected exclusively using molecular techniques and for which no culture is produced and no illnesses detected. And this is the same issue that we're having with cyclospora right now, although the nuances of the cyclospora issue are different than the nuances with the berry issue. I have one last slide that was produced by my colleagues at AFI, which talks about some of the research agendas that could be addressed in the future to answer some of the questions we've brought up. And with that, I will close and I'll be happy to take any questions. Thank you.
Wonderful. Thank you so much for that great presentation.